Hi everyone, in this video I'm going to talk to you about the quadrigeminal cistern and the roof of the third ventricle, also named velum interpositum. On the MR images T2 enhanced, the red arrows are pointing at dilatations with a hyperintense signal content. The left bottom image corresponds to a necropsy of a sheep that presented a cyst damaging the rostral cerebellar lobe. In this video, we are going to analyze through a series of dissections the areas marked with a red and orange arrows on the image. So, as you see, this is a mid-plane section of an encephalon of a dog, and we are going to focus in these areas, the red one and the blue one. And in the red one is where the quadrigeminal cistern is, and the blue one is where the roof of the third ventricle is located, also named velum interpositum. So now you see on the bottom left, you see this midplane section of the encephalon, and this line marks the section that is shown on the right. You see it's at the level of the mesencephalon and pons, okay? And we have divided this in three areas, A, B, C. The A, we can identify the quadrigeminal cistern that is in the subarachnoid space. And in the B and C, we identify also the subarachnoid space with these strands of fibers that are the ambient cisterns on the lateral aspects of the mesencephalon. This drawing represents where the quadrigeminal cistern is located. So it will be on top of the tectum mesencephaly and in between the cerebral hemispheres. You see that it is full of strands of the subarachnoid space, fibers inside. Is where the asterisk is located. And on both sides of the mesencephalon, there are the ambient cistern. So now we go to a midplane section, and as you see, this is taken at the level of the mesencephalon and cerebellum on the right. And you see the mesencephalic aqueduct with the white opening caudally and the tectum mesencephaly. If you follow the tectum mesencephaly, you will identify the rostral medullary velum, partially closing the caudal opening of the mesencephalic aqueduct. And as you see these areas, where you see this arachnoid and all trabeculae that are located in the subarachnoid space. Now these images, the A, the B, the C and the D are dorsal views, where you can see in image A, you see the cerebellum, and rostral, you see the tectum mesencephaly, and in between you have the subarachnoid space filled with these trabecula, with these strands of fibers. What we are going to do is to remove this trabecula that belong to the subarachnoid space, and we are going to move them rostrally. So on figure C, you can see these arachnoid trabeculae that are forming the quadrigeminal cistern that are beginning to be moved rostrally by the forceps. On image D, you can identify when the cerebellum is pushed backwards. You can see the rostral medullary velum, caudal colliculi, and in there you can identify the decussation of the trochlear nerves. So now we are moving these trabeculae of subarachnoid space rostrally. And as we move them, we can reach the epithalamus. You identify the epithalamus, the abenular commissure, both abenuli, and the pineal gland. And look, in image C, you can see when we move with the forceps the arachnoid, we are also moving the pineal gland. In image D, the arachnoid is completely removed, so we identify the pineal gland. So notice that the pineal gland is surrounded by arachnoid. As you see, these are transverse sections. This is a Bialchowski, this is a silver impregnation. On the one on the left, you can identify the subarachnoid space on top of the tectum mesencephaly. And on the right one, you identify the pineal gland, okay, that is surrounded by a lot of vessels included in this subarachnoid space. Now we go back to transverse sections, and on figure A, you see that on top of the tectum mesencephaly, you have this arachnoid that is forming the quadrigeminal cistern, that are a mass of strands of fibers in the subarachnoid space. So CSF can accumulate there, giving rise to cysts inside the quadrigeminal cistern. But on figure B, on the right side, you see that if we move this quadrigeminal cistern and we move this arachnoid, we identify that at the end, we see a membrane that is arachnoid, that serves as a separation, as a wall that separates the quadrigeminal cistern from the third ventricle. Here is a midplane section, so not really a midplane because you can identify the remaining of the parahippocampal gyrus of the left side on the right side, but it's mostly at the midplane. So you see the cerebellum, the tectum mesencephaly, and as you see this arachnoid membrane that extends towards the top of the mesencephalon, and in there it forms this collection of trabeculae that form the quadrigeminal cistern. You can see on the image on the bottom right, you can see all these cavities, okay, that are in the quadrigeminal cistern that may be filled with CSF, giving rise to quadrigeminal cyst. Now, what we do with the forceps is to move up this structure that contains the straight sinus and the great cerebral vein. And as you see, focus on the quadrigeminal cistern, and you will see that rostral to the quadrigeminal cistern, there is this arachnoid membrane that separates the quadrigeminal cistern from the third ventricle that is located rostrally to the quadrigeminal cistern. We also identify the pineal gland enveloped by the arachnoid. On the image on the bottom left, you can identify with one asterisk, is the great cerebral vein of Galenus vein, okay, that is joined by the vein of the corpus callosum, and together they form, on top of the tectum mesencephaly, they are going to form the straight sinus. So now this is an MRI midplane section, and what I want you to show you is this area on the image that is enlarged on the bottom aspect of the screen, so you can identify the third ventricle. This is a T2 enhanced sequence, and also you can identify the velum interpositum or roof of the third ventricle. This is marked with red arrows and the third ventricle with blue arrows. And as you see on top of the tectum mesencephaly, this hyperintense signal triangular shape is the quadrigeminal cistern. you have seen the quadrigeminal cistern in the previous image. On the image on the bottom right, you identify this is at the level of the interthalamic adhesion, and you can identify the third ventricle. This is the blue arrow, and the red arrows are marking the roof of the third ventricle or velum interpositum that contains the choroid plexus of the third ventricle and the internal cerebral veins. In this image, this is a sagittal section, MRI T2 enhanced. So you can identify the interventricular foramen here on the image located on the top right, okay, and is marked with a red arrow. And on the bottom images, you identify the interventricular foramen, and you can compare an MRI with a dissection. So now we make a mid or a sagittal section better. So you can identify here, you can see at the level of the interthalamic adhesion is this section taken. So you see the velum interpositum or roof of the third ventricle and the third ventricle. Also, it is easy to identify the interventricular foramen. So with the forceps, we are moving down the roof of the third ventricle, and you can see that on top was located the fornix and the dentate gyrus and the parahippocampal gyrus. So now we go back to transverse sections and one midplane section. On the image A, this is taken at the level of the methithalamus, okay? And in this area, rectangular area has been enlarged on image B and C. Image B is a rostral vision from rostral to caudal. So you can identify the roof of the third ventricle, and in the middle we see this membrane, this arachnoid membrane that is separating the third ventricle from the quadrigeminal cistern. On image C is a caudal vision. So you can see this arachnoid membrane, okay, marked with a red asterisk, okay, that separates the third ventricle from the quadrigeminal cistern. Don't miss this pineal gland. But the pineal gland here, it is covered by this arachnoid membrane as we have seen in previous images. So this pineal gland is a section, has been sectioned transversally. It is not located caudal to this membrane. It's located rostral, okay? But it's covered by this membrane. So on image D, we identify this arachnoid membrane marked with the red arrow that is separating, okay, the quadrigeminal cistern from the third ventricle. And remember that this membrane extends and covers the pineal gland, because the pineal gland is very highly vascularized. So all these vessels are in the subarachnoid space. So on image A, what do we see? We see with the forceps, we are taking the roof of the third ventricle. As you see, it extends dorsal to the thalamus, okay? And dorsal to the avenula and the pineal gland. Identify that on top of the velum interpositum or roof of the third ventricle, we have the dentate gyrus and the fornix. On image B, we have taken with the forceps this roof of the third ventricle, and we have moved it dorsally, so we see the third ventricle, and also these yellow structures are the choroid plexus of the third ventricle. On image C and D is a transverse section, so you can see the corpus callosum and the commissure of the fornix underneath, and this area, that is whitish area underneath the commissure of the fornix is the roof of the third ventricle that can be separated ventrally from the fornix. This is in image D. It is also easy to identify the choroid plexus of the lateral ventricles. Now with a video and with the forceps, what we are going to do is to move the roof of the third ventricle dorsally. And as you see, it's a very thin membrane that contains the choroid plexus of the third ventricle inside. So now, in image A, this is a midplane section, and this area marked there, we are going to look in detail. So in image B, with the forceps, I am picking the roof of the third ventricle, okay? So in image C, this is very interesting, and image D as well, because you see with the forceps, we are moving down a very thin membrane that is covering underneath the fornix and the dentate gyrus and parahippocampal gyrus, and is located between the parenchyma that is covered by pia mater and the roof of the third ventricle. This thin membrane has been marked with red arrows, as well as in image D. And this is an arachnoid membrane that is located between the roof of the third ventricle and the pia mater that is attached to the parenchyma of the dentate gyrus and fornix. So here you can identify an image A as a midplane section, and we are going to identify these vessels that are running under the dentate gyrus and under the fornix and dorsal to the thalamus, epithalamus, and tectum mesencephaly. So we are going to make a transverse section, as you see in image B, and we are going to have a look from caudal to rostral as the blue arrow indicates in image A. So we go to image C and D, and then you see the abenula clearly, and as you see under this is the hippocampal formation, and in particular is the dentate gyrus. You can see the roof of the third ventricle and the third ventricle underneath, so top of the abenula and under the roof of the third ventricle. And if with the forceps we move down this roof of the third ventricle, we can identify that inside this roof of the third ventricle are located the internal cerebral veins that connect both of them. They form the great cerebral vein or vein of Galen that is easy to see on image C. So for a better understanding of this roof of the third ventricle, imagine during the development. On the figure on the left, you can see the lateral ventricles and the third ventricle. Okay, so the roof of the third ventricle and the mid portions of the lateral ventricles become very thin. These are pia mater and ependymal cell layer. So as the telencephalic vesicles grow, they join at the midplane. So they close this space that is what it was first open dorsally during the development on dorsal portion of the third ventricle. They close them. And what do we have inside? We are going to have that is trapped in this cavity, as you see on the drawing on the right. You can see trapped in this cavity, we have the internal cerebral veins and the choroid plexus of the third ventricle with the rostral choroid artery. The rostral choroid artery is marked in red. So this cavity that it was formed by a folding of the pia, it is the roof of the third ventricle, also named cavum velum interpositum. So it's a double layer of pia. And on top you are going to have the arachnoid, as we have seen on the dissections. So as you see, so you can identify in this image, marked with the asterisk and with the black arrow, the cavum velum interpositum, or roof of the third ventricle. Okay, and that contains the internal cerebral veins and the choroid plexus of the third ventricle. So it may be that an accumulation of CSF due to a malformation during development may cause a cystic dilatation of the cavum velum interpositum. Or maybe an arachnoid cyst pathological may develop there and affect the velum interpositum. These are two different situations. And now you can identify in this dorsal view, you have both thalami, and here I have opened the roof of the third ventricle so we can identify the internal cerebral veins, and running very close to the veins are the choroid plexus. Notice how the internal cerebral veins are joining in order to form the great cerebral vein or vein of Galen. On MRIs on the right, you can identify the choroid plexus of the lateral ventricles, and there in the velum interpositum, the internal cerebral veins and choroid plexus, okay, of the third ventricle. And the MRI that is on the bottom right is taken mucodally, so you can identify now the great cerebral vein or vein of Galen. So to understand that, this is a corrosion cast. In this case was from a cat. So you can see the vein of the corpus callosum marked with number 1, and marked with number 2 is the internal cerebral veins that join in order to form the great cerebral vein or vein of Galen. Is number 3. They join the vein of the corpus callosum and they form the straight sinus that is marked with number 5. These are joined by the dorsal sagittal sinus, and they form the communication between the dorsal sagittal sinus and the vein of Galen. This communication is shown on the image on the top right. So if we move to the bottom images, we enlarge this and we identify the internal cerebral veins that they joined in order to form the vein of Galen or great cerebral vein that is located on top of the tectum mesencephaly. So you see here, you see the internal cerebral veins, they join in order to form the great cerebral vein, and they follow as the straight sinus. On transverse sections T2 and haste, we are going to look at the areas enlarged at the bottom of the image. Section A is more rostral than section B. On section A, we can identify the internal cerebral veins with the 2 rows of the choroid plexuses of the third ventricle on the roof of the third ventricle. On section B, the 2 internal cerebral veins have joined to become the great cerebral vein or vein of Galen, and we still identify the third ventricle. Here you see is a midplane section MRI image. You see the internal cerebral veins, how they join to form the great cerebral vein, and this goes up in order to form the straight sinus. On the bottom images, you can identify the great cerebral vein, okay, and on the one on the right, you see the roof of the third ventricle that has been opened, okay, that is very thin. Remember, is this double layer of pia, the roof that contains the internal cerebral veins and the choroid plexus of the third ventricle together with the rostral choroid arteries. Here we have a video of a dissection of a dog's encephalon. I have removed part of the left cerebral hemisphere in order to identify here with the forceps the two internal cerebral veins that run with the choroid plexuses of the third ventricle. Both veins join in order to form the great cerebral vein or vein of Galen. That's it. And this is joined by the vein of the corpus callosum to form the straight sinus. This is it. Thanks for your attention.